And here we are, Chapter 8, Microbial Genetics. This will be the last chapter for week 1. So when we're talking about bacterial genetics, we're talking about genes. And genetics is the study of the genes, how do are they expressed, things of that nature. But the gene itself is DNA. And what's happening with the DNA is that you are able to encode information like a blueprint, and that's called a gene. So a collection of the genes are on this circular piece of DNA in bacteria. And this is showing you some of the chromosomes or actually the chromosome. And this is showing you the flow of genetic information. So with the flow of genetic information, what you have on the left-hand side, the first column, is a bacterial cell with a circular piece of DNA. And we'll get into recombination later, but right now what's happening is that the cell is about ready to divide. It's gotten large enough. And so in the middle, what you have is the replication of the circular chromosome. So each of the daughter cells after the division have an exact copy. And what then happens is that the cell wants to express itself. So in order to make a protein, structural enzyme, it's going to have to go back to the gene on the DNA, make a copy. Messenger RNA is the copy, and the process is called transcription. This is on the right-hand column. And then the messenger RNA is able to go to the area in the cell where there is ribosomes, amino acids, ATP, and then they are able to actually use that blueprint, read that messenger RNA, and make a structural protein. And here is a double helix of DNA, and what you're seeing right now is it's beginning to replicate and make a copy of itself. But I'm just going to be going over the general process. So you realize that you are going to have your A's and your T's and your G's and your C's, your nucleotide that actually are able to code for amino acids that then can be linked into making a protein. And if or when you are making a copy of the instruction tape of the DNA and you're doing DNA replication, the enzyme that I do want you to know that is so important in allowing the structure to replicate itself and make a perfect copy is DNA polymerase. So it says DNA contains all the information, so it has to be copied prior to cell division. In this process, DNA polymerase copies the circular piece of DNA until 2 copies are formed. And then after binary fission, each cell will have a full set of instructions. Now, with the between cell division, this is when you're going to be doing the protein synthesis. A cell, whether it's bacteria or your own cells, have to express themselves. They have to make things. They have to grow. They have to make toxins or other types of signal molecules. And this is the time that they're going to do it. They're going to do it between divisions. So as between cell divisions, the bacteria need to make proteins in order to build and survive. This is a multi-stage process, beginning with the copying of the DNA instructions by RNA polymerase. Note that. To make messenger RNA, note that as well. This is called transcription. To transcribe means to copy. So once you transcribe and make your messenger RNA, then you're going to go to that area in the bacterial cell where the messenger RNA can bind to ribosomes and be read to form a protein. And this protein product is done through a process called protein synthesis. So this is actually an electron micrograph where you can see the RNA polymerase making a copy the messenger RNA from the DNA template. So, and then once the RNA is made, it then can undergo translation and the copy can be read. But this is transcription. So your messenger RNA is translated. We realize that it's read like a sentence, but the sentences are composed of words that are only 3 letters or 3 nucleotides long. So it would be like see cat run. And that is, the sentence would be like a protein and the individual words would be the amino acids. So the says a messenger RNA is translated in codons, 3 nucleotides. Translation of the messenger RNA begins at a start codon. Don't memorize AUG, but just know the process that you're going to start, then you're going to proceed. And I'd like to think of putting on the amino acids according to the messenger RNA, like putting beads on a necklace. And then once you're done, you have to tell the cell that yes, you're done, stop. And then you do have a stop codon. Don't memorize the stop codons, but just realize that you have a beginning, then you proceed to the end. And at the very end, you have yourself a protein. So the transcription is the copying of the DNA blueprint, and then the translation is reading that blueprint to make a protein. And that's it. So that will be it for week 1, and I will see you soon for week 2.